we talked about operons. That's how bacteria change their gene expression on a moment-to-moment basis. This is happening in the environment now. I change my genes now. Environment changes 2 minutes later. I change my gene expression 2 minutes later. Okay, so it's a moment-to-moment adjustment of gene expression. Epigenetics is more permanent. An epigenetic shift in gene expression can last an organism's entire lifetime. It can even be inherited by future generations. So this epigenetics explains why your muscle cells are different from your heart cells and why they are different your whole life in that way. It also explains why bacteria behave differently and look different in biofilms versus when they're free-floating. Okay, so this is gene expression, setting a programming of gene expression that can last the organism's entire lifetime and can even be inherited by future generations. So epigenetics is when the environment and the experiences of the parent, either the parent organism or the parent cell, alters how genes of daughter cells or offspring are expressed. And usually it's because of DNA methylation. So there's the genetic code. The genetic code is not being changed, but you can add a methyl group. Methyl is just a carbon surrounded by a few hydrogens. When you methylate the DNA, when you add a methyl group to the beginning of a gene, it turns that gene off. That methyl group is covalently bound, unlike the repressor. So the repressor in the lac operon, that can be removed and added back on. A methyl group is a semi-permanent alteration of the gene. It's not changing the genetic code. If you were to sequence that DNA, you wouldn't see if it was on or off, you would just see the code, right? The code is the same. It's not a mutation. The genetic code stays the same. We're talking about a methyl group, a molecule that is covalently bound to the outside of the DNA that cannot be removed. And actually, we can do epigenetic sequencing, where we can read which genes are methylated and which are not. So when a gene is methylated, it is turned off. It cannot be transcribed or translated, okay? So several studies, my diagram here is these giraffes, because this is a little bit like Lamarckian evolution. So Darwin is always credited with the idea of evolution. In truth, the idea had been coming about in the sciences for a long time, because scientists read each other's work. It's a collaborative community. Everybody's mind starts to be changed by the data simultaneously. Darwin is just the first person who put together a cohesive explanation of evolution in a book first. Okay, so he gets credited. Lots of people were coming to the same conclusions at the same time, and actually he didn't come up with evolution. The idea that organisms change over time actually was written down earlier by a guy named Lamarck. Lamarck had the idea first that organisms weren't just static, that they could change over time, but he didn't have it right exactly how it worked. So in his mind, his famous example is giraffes. So his explanation of how giraffes' necks get longer and got to be so long is that you had these animals that were sort of like giraffes but had shorter necks, and they lived in the savannas, and they were eating the leaves off the trees, and so the leaves that remained were higher and higher and harder to reach. The giraffes had to stretch their necks to reach the higher leaves, and their necks got a little bit longer. And because their necks stretched out, their offspring's necks were a little bit longer. And so every generation of giraffes, they were eating the leaves higher and higher up. They needed to stretch further and further to reach the available leaves, and they stretched their necks, their offspring inherited the stretched neck. Now, we know that this doesn't work anymore, right? Because here it says stretch, think about the children. Right? Make that neck longer so our children will be born with a long neck. So if this were true, that would sort of be like saying if your hand got lobbed off, your child would be born without a hand. And we know that that's not how evolution works. That is not how change is inherited. But there are some elements of this that it now turns out are true. We can add some elements of this to Lamarckian evolution. So it turns out that our health, if we're talking about humans, there's a lot of really interesting research right now in epigenetics and its implications on metabolic disorders and intergenerational cycles of drug addiction and intergenerational cycles of violence. So let's talk about the metabolism first. So several studies have shown that a poor or high-fat diet is detrimental to health across several generations. Now, epigenetic factors have also been implicated in the transgenerational effects of such an inappropriate diet, meaning that we now know that DNA methylation, we know that when a parent has a really bad diet, they get a metabolic disorder, their epigenetics change. The methylation, which genes are on or off, gets changed, and then that epigenetic change, the DNA methylation, gets passed on to offspring. So the position of those methyl groups, the on-off position of the genes, can be inherited by offspring. Now, these effects arise both through maternal and paternal transmission. So we're always saying, well, a mother needs to prepare her body for pregnancy. She needs to get healthy. She needs to create a good uterine environment for that child. Well, because of epigenetics, it turns out that men can affect the health of their offspring through their own health as well. And the studies on this are becoming incredibly robust. When we study epigenetics, we study it in men because you want to take out the effects of the uterus, right? So if you're studying mice and you're studying metabolic disorders in mice, you don't want to use a female mouse because if the female has a metabolic disorder and then she carries her baby mice in her uterus, you might think, well, they're inheriting this metabolic disorder because they were in this toxic uterine environment or something like that, right? So what you do is you study it through men because the men just give the sperm. And so any effects of transgenerational effects from men to offspring, that's not because of the uterine environment, that's because of epigenetics, okay? So what they find is in rats, in the lab, they feed these male rats a really high-fat diet. Those males generated offspring with diabetes-like conditions that were manifested by impaired glucose tolerance and insulin secretion, even though the offspring were fed a normal diet. So the male mice, they were fed a high-fat diet until they developed metabolic disorders. Then they mated those male mice with female mice that were fed a normal diet and did not have metabolic disorders. And they found that the offspring of the male mice who were fed a high-fat diet, their offspring also had metabolic disorders even though they had a normal diet and the mothers had a normal diet. Okay, so and then not only that, but the metabolic disorders in the offspring were associated with abnormal gene expression in pancreatic cells of the offspring, and that was correlated with DNA methylation of certain metabolic genes. So the fathers were fed a high-fat diet, their epigenetics changed to adapt them to this diet, and then the epigenetic profiles were passed to their offspring. Okay. And so I can give a link to that research paper, but there's lots of research papers like that. So for example, another rat study, they take these, again, male rats, and they expose them to the sweet almond scent. It's an olfactory molecule. It binds into receptors in the olfactory system so that they can smell this sweet almond scent. And every time they expose them to this scent, they shock them with an electric shock. Now they do this several times a day for several weeks. Then they mate these rats with female rats that have not experienced this trauma. The offspring have not received this trauma, and yet they are hypersensitive to that olfactory molecule. They are very, very sensitive to that scent. It's not that they inherit fear of that scent, they are just hypersensitive to it because they produce more receptors in their olfactory system, allowing them to be more sensitive. They have more receptors to bind to that scent molecule so that they can detect smaller quantities of it. Okay. Now, the whole idea, the purpose of epigenetics is to prepare the offspring so the parents can prepare the offspring for the type of environment they will encounter. Right? So when you think about mammals, what the parents experience, usually the environment doesn't change that fast. So usually the stresses experienced by the parents are going to be similar to the stresses experienced by the offspring. So it is, yes, they have their genetic programming, which has been affected by evolution. Only the best genes were passed on to the next generation, of course. But in addition to that, there's this ability for the parent, it's not conscious, it's just this DNA programming, this DNA methylation. So the environment causes certain genes to be turned on and other genes to be turned off, and the on position of those genes can be passed to offspring to prepare them for the kind of stresses that they will encounter. So if rats are going to be, the idea is if these rats are going to be in danger every time they smell that scent, they should be able to smell it better so that they're better able to avoid danger. right? Now, not all, no, so all of us experience epigenetic changes in our lifetime to our genome, to our epigenome. Not all of those epigenetic programs get passed to offspring. Some of it gets cleared, some of it doesn't, and we don't exactly know why yet, but we do know for sure that some of it gets passed to offspring. Okay, so the reason this is implicated in things like intergenerational cycles of addiction and violence is because your stress response system is programmed epigenetically in utero and in early childhood. So the stresses encountered, you know, the mother's stress, so the mother's stress hormones are shared with her baby while the baby is in utero, so that counts too. So maternal stress and also stresses encountered by a child in early childhood. Early childhood is where epigenetic so you can think of children, young children, as hot wax. The things they experience are molding their epigenome, and at some point the epigenome stops changing very easily. But in early childhood, these programs are being set. The child is experiencing the stresses of the environment, and genes are being programmed accordingly. So the genes that are that are responsible for the production of stress hormones and adrenaline, you know, things like that, that's our hypothalamic pituitary adrenal axis. It's our stress response system. It's part of our, that is a piece of our neuroendocrine system. And that gets programmed in response to the stresses we encounter. So if in early childhood you experience a lot of stress, you're going to be programmed, your neuroendocrine system and your stress response system is going to be programmed, your fight and flight system is going to be programmed in such a way to help you survive in high stress situations. Then later in life, when you escape those high stress situations, you're still programmed that way. You don't lose the programming if you do, not very easily in adulthood. And so this is one of the things that's very challenging, is that if you've been born into violence, it's hard to leave the violence behind. It's not impossible. We do find that epigenetics can be reprogrammed, but we still have a lot to learn about how to do that. So if you're interested in the human social implications of epigenetics, you should look into intergenerational cycles of violence and epigenetics. It's really interesting. So in terms of human cells, epigenetics also play a role in defining liver cells from skin cells, from neurons. So we all start as stem cells that have not been assigned a role yet. And once they're assigned a role to be skin or blood or nervous tissue or muscular tissue, they don't go back. The epigenetic programming that says, I'm going to express these skin cell genes and I'm not going to express those nervous system genes, or vice versa, that does not go backwards. Once a cell is programmed to be a certain cell type, it doesn't go back. All your cells have all the genes to be any cell type, but some of them get turned off permanently during development so that you have different tissue types that always stay the correct kind of tissue, doing what they're supposed to do in the cell. All right, so with bacteria, this plays a role as well, because there are some bacteria that live peacefully in your body. If you encounter a certain stress, or they encounter a certain stress, they switch on pathogen genes, and then they become pathogenic, and their offspring will be pathogenic. Then they can switch those off again at a later date because of different environmental stimuli. So this is another way, you know, again, like I said before, there is the nature versus nurture debate is stupid. Epigenetics, there is no dichotomy. It is both. It is 100% always going to be both. So your genetics set the range of expressions that you can have. You know, like, you're never going to be a dog, right? So there's a range of ways that you can express your hormones, your structure, your body, your brain, but then the environment. But within that range, the environment plays a huge role. So the brain is very plastic. We know that the brain, IQ and intelligence is approximately as much influenced by environment as it is by genetics. Okay, so we're not set in stone. Our early environment, especially, programs who we're going to be later in life. Just one last epigenetic control method. I think your quiz might ask you for two. and I can't remember what's in this version of the textbook off the top of my head, so I'm going to list another one. It doesn't matter for bacteria, but you should know that it exists. So when you add a methyl group, that's DNA methylation, that turns off genes for the long term. There's also histone acetylation. Histone acetylation. Bacteria don't have histones, but archaea and eukaryota do. Histones are proteins that the DNA wraps around to stay organized, okay? So you can think of like you keep your thread organized, right? You don't just have like loose thread floating around in your thread basket, your sewing basket, because that would get all tangled, be a big tangled mess. What you do is you wrap the thread around a spool. Histones are proteins that act as spools to wrap the DNA around to keep it organized. Now, when the histones are have an acetyl group attached to them, they wrap the DNA more tightly. The tighter the DNA is wrapped, the less accessible the genes are that it contains. So think about the spool of thread. If you wrap thread around a spool, you have to unravel it to use it for sewing, right? You can't use the like that thread that's wrapped tightly around the spool on the inside of the thread, in the inside of the spool. It's not accessible to you. You have to unwind the thread from the outside to use it for sewing. So it's the same thing here. So if you acetylate the histones, the DNA is going to wrap around those histones tighter, and the genes contained there will not be accessible. They will be turned off. So you have DNA methylation, bacteria, archaea, eukaryota. We all use DNA methylation to turn genes on or off for the long term. But also there's acetyl groups, and acetyl groups affect histone wrapping. So that's just for archaea and eukaryota, which have histones.